I want you to picture a scene with me for a second. It's something straight out of a misty, classic Gothic novel.
Oh, I love where this is going.
Right. So you're walking through a chilly, damp graveyard. The air is really heavy, and there's condensation clinging to all the old headstones.
Very atmospheric.
Yeah, and right there, sprouting literally right out of the recently disturbed earth, sits this somber, woolly little mushroom.
Just minding its own business in the dirt.
Exactly, but if you lean in close, you'll see that it is literally weeping. It's exuding these, like, pitch black tears from its edges, just dripping down into the soil.
It is such a wild visual. It really is.
It's so creepy. You might look at it and think it's just some weird forest oddity, you know? But hidden inside this weeping fungus is an unresolved psychedelic mystery and, incredibly, a molecular bloodhound that is actively changing modern oncology.
Which is massive.
Welcome to our deep dive into the Weeping Widow. Our mission today is to take a huge stack of mycological papers, field guides, and complex biochemical studies and figure out how a dark, bitter-tasting mushroom crying in a cemetery actually holds the blueprint to hunting down human cancer.
It's really the ultimate testament to why basic biodiversity research matters so much.
How so?
Well, the organisms that look the most bizarre or even macabre to us are often the very ones carrying the most profound biochemical tools. I mean, we are looking at a species today that completely defies our traditional systems of classification.
It breaks all the rules.
It breaks the rules of our chemical databases, absolutely. And yet, it provides a level of diagnostic precision that human engineers, even with all our technology, really struggle to replicate in a laboratory.
Okay, let's unpack this.
Yeah.
Because the scientific name really sets the stage perfectly for what we're dealing with here.
It's one of my favorite binomial names, actually.
It's Lacrymaria, lacrymabunda, which is just a mouthful.
It is.
But for anyone rusty on their Latin, both parts of that name originate from the modern Latin word lacrimaris, which pertains to tears.
Right, so the genus name, Lacrymaria, essentially means that which produces tears.
And the specific epithet, lacrymabunda, means abundantly weeping.
So put it all together, and the scientific community officially named this thing the abundantly weeping tear producer.
They really leaned into the goth vibe.
They did. Mycologists are nothing if not descriptive. And, commonly it's just called the weeping widow.
Which is such a great common name. But let's talk about what this thing actually looks like if you stumble across it.
Yeah, so macroscopically, meaning what you can see just walking past it on a trail, it's a reasonably sized mushroom.
Not microscopic, but not huge either.
Exactly. The cap usually ranges from maybe 4 to 12 centimeters across.
It's about the size of the palm of your hand, maybe a bit smaller.
Yeah, exactly. And when it first pushes through the soil, it's shaped like a tight little bell. But as it matures, that cap expands out, becoming broadly convex, so almost flat, but it retains a distinct raised bump right in the center.
Right. Mycologists call that structure an umbo, right?
An umbo, yes. It's a very specific architectural feature.
To me, it looks like the boss on a Viking shield. You know, that metal bump in the middle.
Oh, that's a perfect comparison, yes.
But it's the texture that really gives it that disheveled widow and mourning vibe we were talking about.
The shagginess.
Yeah, the sources describe the cap as being covered in these reddish to yellow-brown, sometimes kind of clay-brown, hair-like fibers.
It's densely fibrillose is the technical term.
Fibrous. It basically just looks woolly and unkempt. And hanging off the very edge of that cap, what they call the margin, you usually find these white, cotton-like remnants of a partial veil.
Which is actually super important structurally. That partial veil serves a critical protective function when the fruiting body is young.
Like a blanket.
Sort of, yeah. It covers the developing gills underneath, shielding the really immature, delicate, spore-producing tissues from drying out or, from being eaten by microarthropods in the soil.
Bugs love to eat baby mushrooms.
They absolutely do. So as the cap expands outward, that veil physically tears under the tension, and it leaves this delicate, ragged white fringe hanging down all around the perimeter.
Adding to the whole tattered widow aesthetic.
Exactly. And below that, the stem is hollow and features a really distinct black ring near the top.
But the ring isn't naturally black, is it?
No, it's actually stained black by the falling spores, which kind of hints at the main event happening underneath.
The weeping.
The weeping, yes. The guttation.
Guttation. Let's really dig into this because this is the biological process that creates those famous black tears.
It's fascinating to watch in person.
I bet. So in moist conditions, especially when there's high humidity, the gills of this mushroom actively exude droplets of liquid.
They just push it right out.
Yeah, and it starts out clear, right? Maybe a little cloudy or just watery.
Just looking like standard morning dew at first.
Right, but as the mushroom matures, the gills start releasing millions of jet black spores.
And those dark spores get caught in the clear droplets, turning them entirely opaque black.
So they literally hang off the fringed edges of the gills like these heavy beads of dark ink. It's visually stunning for us to look at.
It really is.
But from a biological standpoint, I mean, pushing liquid out of your own tissues like that requires energy. It requires mechanical force.
It requires immense internal turgor pressure.
Turgor pressure. Break that down for us.
Sure. So fungi grow by pumping water into their cells,
Yeah.
basically expanding them like elongated water balloons.
Okay, that makes sense.
This turgor pressure is what drives the hyphae, the thread-like structures that actually make up the mushroom forward through the dense soil and then upward into the air to form the cap.
So it's basically a microscopic hydraulic system.
That's exactly what it is.
Yeah.
And guttation is the process where some of that internal liquid is forced all the way to the surface, completely overcoming the surface tension of the cell walls.
Wow. But why? Like, what is the evolutionary reason the weeping widow does this so prolifically?
Well, that is a genuine blank spot in the scientific literature, honestly.
Wait, really? We don't know why it
But we don't have a definitive single answer, no. There are, however, 4 primary hypotheses that researchers are currently untangling.
Okay, let's break those down. Theory number 1 from our sources is entomophilous dispersal, which basically means dispersal by insects.
Right. Bug transit.
Bug transit, yeah.
Yeah.
The idea here is that these droplets aren't just pure water, they're a cocktail.
A very intentional cocktail. They likely contain dissolved proteins, perhaps trace sugars.
Right, so if you are a fly or a gnat navigating a damp field, a droplet of nutrient-rich liquid is a major resource.
They have an energy drink just sitting there.
Exactly. So the insect lands, drinks the droplet, and in the process gets totally coated in those jet black spores.
And then it flies off to a new location, acting as an unwitting cargo plane for the fungus.
That's so clever.
It is. And bribing insects with a sugary or protein-rich exudate is a well-documented strategy in other biological kingdoms, particularly plants. Think of nectar.
Oh, sure, like bees and flowers.
Exactly. If the droplets are indeed acting as bait, it totally solves the problem of how to disperse heavy spores on days when the wind just isn't blowing.
Okay, but if we jump to theory number 2, it has nothing to do with bugs at all. It's about water regulation.
Back to the hydraulics.
Yeah, we just talked about that turgor pressure. If a mushroom is growing incredibly fast overnight, maybe it needs a pressure relief valve.
Because the pressure can get too high.
Right. By actively sweating out excess water, the fungus can maintain a constant optimal internal hydraulic pressure.
Which will allow the aerial hyphae of the fruiting body to keep expanding without actually bursting.
Bursting would be bad.
Very bad. Think of the sheer volume of water being moved from the underground mycelial network all the way up into the fruiting body.
It's a massive plumbing effort for a little organism.
It is. And if the ambient humidity in the air suddenly spikes, say it starts raining, evaporation from the mushroom's surface slows way down.
Oh, so the water has nowhere to go.
Exactly. The internal pressure could become dangerously high, so guttation might simply be an active mechanical exhaust system to prevent catastrophic cellular rupture.
That makes total sense. Which leads right into theory 3, metabolic cleansing.
Flushing the system.
Yeah. Growing a complex structure like a whole mushroom cap overnight generates a lot of metabolic waste. I mean, our bodies flush waste through our kidneys.
Right.
So this hypothesis suggests that guttation droplets are essentially a vehicle to remove toxic byproducts that get generated during the rapid cell division of the fruiting body.
The fungus is literally flushing its trash.
Pushing the trash out into these external droplets so it doesn't poison its own tissues as it grows.
It's very elegant. And
Yeah.
Theory 4 takes that concept of pushing out chemicals and weaponizes it.
Chemical defense.
Exactly. Chemical warfare. We see this in other gutating fungi, like the bleeding tooth fungus, where the exudates are just packed with deterrents.
Because a mushroom is basically just a highly nutritious package of proteins and fats, right?
Right, and it's completely defenseless against grazing slugs or antagonistic molds.
Yeah.
It can't run away.
So it is highly probable that the weeping widow is pumping these droplets full of antimicrobial compounds or, anti-herbivore toxins.
Deploying a chemical moat around its reproductive organs.
A chemical moat. I love that.
Uh-huh.
But I have to say, I want to challenge that first theory about the insects for a second.
Okay, let's hear it.
If this is just about attracting a fly with a sugary drink, why are the droplets forming specifically on the delicate gill edges? Right, because if you want to attract bugs, wouldn't it be far more efficient to just sweat those nutrients all over the top of the broad cap?
Make a bigger target?
Yeah, it's a huge landing pad up there. Sweating only from the very edge of the gills underneath feels less like a full-body cooling system or a billboard for bugs, and much more like a highly targeted, localized process.
It's very precise.
It's like weeping from a specific wound rather than sweating from your skin.
That localization is the key to the whole puzzle, honestly. You are pointing out a phenomenon that requires intense tissue specialization.
How do you mean?
Well, the cells at the very edge of the gills, the lamellar margin, are structurally and functionally distinct from the cells on the face of the gills, or the top of the cap.
They're built differently.
Exactly. The fact that the liquid is exuded exactly at the precipice where the mature spores are actively being released implies a deliberate mechanical interaction.
So they're working together.
Right. It might be to prevent the gills from drying out and fusing together before the spores can catch the wind, or it might be to anchor those spores in a microenvironment until an insect specifically grazes that exact edge. The biological expense of being so precise is what makes it so evolutionarily significant. Nature doesn't waste energy like that for no reason.
No, it doesn't. And that precision tells us a lot about how it interacts with its environment, which brings us to where this mushroom actually chooses to live.
Habitat is everything.
Because it doesn't just pop up in any old forest, it is a saprotroph.
Right.
So for you listening, in the fungal kingdom, you have mycorrhizal fungi, which are the ones that form those vast mutualistic partnerships with the living roots of trees.
They trade soil minerals for the tree's photosynthesized sugars. It's a beautiful trade agreement.
But then you have saprotrophs. They are the decomposers.
Recyclers.
Yeah. They make their living by secreting powerful enzymes directly into dead organic matter, leaf litter, decaying grass, wood chips, and absorbing the broken down nutrients.
Being a saprotroph means the weeping widow is basically an independent contractor.
It doesn't have a boss.
Exactly. It is completely untethered from the forest canopy. A mycorrhizal fungus, like, say, a chanterelle, will die if its host tree is cut down.
Because the sugar supply gets cut off.
Right.
Yeah.
But lacrymabunda has immense geographical flexibility. It can colonize anywhere. There's decaying organic material and sufficient moisture.
Anywhere.
Pretty much.
But our sources say it has a very specific preference for disturbed, nitrogen-rich ground. The lists include gardens, urban parks, roadside verges, gravel driveways, and notably, graveyards.
It does seem to love a cemetery.
But why disturbed ground? I mean, if you've ever dug up a garden, you know you're just moving dirt around. What makes a freshly landscaped park or a newly dug grave such a paradise for this specific mushroom?
It is entirely a matter of ecological niches and outrunning the competition.
Outrunning?
Yeah. So think about an ancient, undisturbed forest floor. That is a saturated market. Every microscopic grain of soil is heavily contested territory.
Really?
Oh, yeah. Thousands of species of established fungi, bacteria, nematodes, they are all locked in a perpetual turf war for resources.
So a new spore landing there has very little chance of establishing a foothold.
Almost zero.
So the established microbial community basically bullies the newcomers.
Precisely. They produce antibiotics to kill competitors. But human activity changes the math completely.
By disturbing the soil.
Right. When we till a garden bed, or lay down fresh wood chips in a suburban park, or dig into the earth in a cemetery, we alter the landscape in two profound ways.
Okay, what's the first?
First, we chop up and bury a massive amount of organic matter, grass roots, leaves, topsoil. We create an instant, localized feast.
A buffet just sitting under the surface.
Exactly. And second, the physical disruption wipes out that established microbial hierarchy we just talked about. We essentially clear the board.
Oh, wow. So the weeping widow is just an opportunist sweeping into the vacuum.
It is a first responder. Saprobes are evolutionarily adapted to exploit these sudden influxes of decaying matter.
They like the chaos.
They thrive in it. They grow aggressively in the temporary absence of competition, breaking down the complex lignins and celluloses of all the debris we leave behind. They really thrive in the messy wake of human habitation.
Which is fascinating, but also kind of problematic. Because they live right in our backyards and city parks, people inevitably wonder about throwing them in a frying pan.
Oh, the foraging question.
Yeah, urban foraging is huge right now. People love finding free food. But the culinary consensus on the weeping widow is incredibly bleak.
It is not a gourmet mushroom.
Not at all. Field guides almost universally classify it as not recommended or bluntly inedible.
Yeah.
And it's not necessarily deadly poisonous, right? Like it's not a death cap. But it's just considered a phenomenally bad idea to eat it.
It is profoundly unappetizing.
The texts note that it tastes intensely bitter, and if you try to cook it, the high water content and that massive payload of black spores turn the whole thing into just a black, soggy mess in the pan.
Nobody wants to eat black mush. And beyond the bitter taste and the muddy texture, there is a consistent clinical history of it causing mild to moderate gastrointestinal distress.
Like cramping, bloating, nausea.
Exactly. The human digestive tract clearly flags something in its cellular makeup as an irritant. However, and this is crucial, the intrinsic bitterness isn't actually the primary danger here.
No.
No. The real threat comes directly from its status as an urban opportunist.
Huh.
We have to look at bioaccumulation.
Right. Bioaccumulation. Fungi are essentially nature's sponges.
They are.
The underground mycelium is this vast microscopic net of absorbent threads, and they have to pull water and nutrients from the surrounding soil to survive, obviously.
But they don't have a sophisticated internal filtration system to reject environmental contaminants.
Right, so they absorb their environment completely indiscriminately.
Exactly. When a weeping widow colonizes a roadside verge, its mycelial network is pulling in all the heavy metals settling from car exhaust.
Oh, man.
The zinc and cadmium from tire wear on the asphalt, the petroleum hydrocarbons washing off the road when it rains.
And in a landscape park or a golf course, it is absorbing synthetic fertilizers and chemical pesticides.
All of it. It concentrates these ambient toxins right within the fleshy tissues of its fruiting body.
So it's basically acting as a biological cleanup crew, but it just sweeps all that toxic urban runoff into its own tissue.
That's a great way to visualize it.
Which means if a forager plucks that mushroom thinking they found a snack, they are literally holding a concentrated toxic package.
It is a critical, often totally overlooked principle of foraging. A mushroom's edibility is inextricably tied to its habitat.
Right, because you could find a universally prized edible species, like a porcini. But if you harvest it from an old industrial site or a heavily sprayed suburban lawn...
You are ingesting the legacy of that pollution?
Wow.
The weeping widow is the perfect case study for this. Even if it tasted wonderful, its ecological preference for the disturbed, polluted margins of human activity makes it inherently hazardous.
You are eating the chemical history of the soil.
Exactly.
It's a very sobering reality check for anyone hunting for dinner in a city park.
Definitely stick to the grocery store if you aren't sure about the soil history.
For real. Now, we need to pivot, because this mushroom's visual weirdness and habitat are completely matched by the absolute chaos it caused in the scientific community.
Oh, the taxonomic turmoil.
The taxonomic history of the weeping widow is a nightmare of indecision. Back in 1785, it was originally classified as Agaricus lacrymabundus.
Right.
Then scientists just started shuffling it around. It spent decades bouncing between names like Psathyrella velutina and Lacrymaria velutina. They just could not agree on what family tree this thing actually belonged to.
And you really can't blame them because the confusion stems entirely from the limitations of historical taxonomy.
Before we had fancy lab equipment.
Right. For centuries, mycology relied almost exclusively on macroscopic morphology.
Which just means what it looks like to the naked eye.
Exactly. Scientists looked at the visible architecture, the shape of the cap, the color of the spores, how the gills attached to the stem, and they grouped organisms that looked similar.
Seems logical enough.
It does, until you realize nature is full of copycats. The weeping widow is basically a small to medium brown mushroom with a fragile cap and dark spores.
Which describes, like, half the mushrooms in the forest.
Right. So for a very long time, the genus Psathyrella served as a polyphyletic dumping ground for mushrooms matching that vague description.
A polyphyletic group. Let's really unpack that, because it's crucial to understanding how molecular biology changed everything later on.
Please do.
So, in evolutionary biology, a monophyletic group is a true family tree. It includes a single common ancestor and every single descendant that evolved from it.
Like humans and all other apes sharing a common primate ancestor.
Exactly. But a polyphyletic group is artificial. It's a collection of organisms grouped together because they happen to share similar physical traits, but they don't actually share a recent common ancestor.
Often due to convergent evolution.
Right, meaning they independently evolved similar solutions to similar environmental problems.
It is the biological equivalent of organizing a massive, cluttered closet by simply taking everything that is brown and fragile and throwing it into one big box labeled Psathyrella.
Without looking at what the items actually are.
Exactly. Regardless of whether the item is a wool sweater, a delicate teacup, or a pair of leather shoes.
They all go in the brown, fragile box.
Right. It looks organized on the outside, but structurally, the items have absolutely no relation to each other. The weeping widow was tossed into that box and left there for years.
But then, in 2008, a research team led by Padamsey published a study that completely shattered that old system using molecular phylogenetics.
The DNA revolution.
They stopped looking at the shape of the mushroom and started looking at the actual source code. They sequenced the DNA.
And what they did was target specific regions of ribosomal DNA across dozens of these little brown mushroom species.
Looking for the real family ties.
By comparing the exact sequences of base pairs, they could finally see the true genetic divergence. And the data proved conclusively that the Psathyrella genus was a tangled mess of unrelated evolutionary lineages.
The box was full of random junk.
Exactly. The genetic evidence provided the legal and scientific mandate to separate the weeping widow, along with a few close relatives, out of that artificial grouping and elevate them into their own distinct genus, Lacrymaria.
But, even before we had the technology to sequence DNA, there were early mycologists arguing that it didn't belong in that Psathyrella box.
They were noticing the subtle clues.
Yeah. Macroscopically, a true Psathyrella mushroom has a hygrophenosis cap.
We're famous.
Right. That means the cap drastically changes color as it loses moisture, fading from a dark brown to a pale tan as it dries out.
Like a wet sponge drying on the counter.
Exactly. But the weeping widow is non-hygrophonous. Because of all those dense, shaggy fibers on the cap, its color remains relatively stable whether it's soaking wet or totally dry.
So that was the first hint that it was an imposter.
Yeah. But the real smoking gun was found when they put in the spores under a microscope. Here's where it gets really interesting.
I love this part.
The sources have this incredible quote from a researcher looking at the spores. They asked, how much more action-packed could a mushroom be?
It's a completely justified level of enthusiasm from a mycologist.
Right. Why are they so action-packed?
Well, if you place a spore from a true Psathyrella under high magnification, it is generally smooth, oval, and pretty featureless.
Like a microscopic jelly bean.
A microscopic jelly bean, yes. But the spore of Lacrymaria, lacrymabunda, is this complex, heavily armored structure.
Armored.
Yeah, it is jet black, and the entire surface is heavily warty and roughened, like a tiny asteroid.
Whoa.
Furthermore, at one apex of the spore, there is a highly distinct, protruding germ pore.
The literature specifically describes it as snout-like.
Snout-like.
It's a warty, microscopic balloon with a distinct snout.
That is so weird. And the microscopic complexity doesn't stop with the spores, does it? Not at all. If you look at the very edge of the gills, the exact location where the guttation droplets form, you find dense clusters of specialized sterile cells.
Chylocystidia.
Yes, chylocystidia. Chylo refers to a margin or a lip, and cystidia are sterile cells that do not produce spores found on the surface of fungal tissues.
So they're just hanging out on the edge.
Right, but in the weeping widow, these chylocystidia are club-shaped, and they feature these dramatically swollen, knob-headed tips.
Like little microscopic drumsticks.
Exactly. And they cluster together so tightly along the lamellar margin that they create a physical microscopic fringe.
So we have a mushroom deploying armored, snouted spores and a dense fringe of knobby sterile cells. How do these microscopic architectural features actually help this organism survive out in the dirt?
It all comes back to environmental adaptation.
Okay, let's start with the spores. Why the warts?
The roughened, warty texture of the spore is very likely an adhesion mechanism. Think about it. A perfectly smooth spore can be easily washed deeper into the soil profile by heavy rain.
And if it gets washed too deep?
It gets buried too deep to ever germinate and reach the surface. But a warty spore has friction. It can snag onto a passing insect's leg, or anchor itself firmly to the specific type of rough, disturbed soil particles it prefers to colonize.
It's like microscopic Velcro.
Exactly. And the snout-like germ pore serves as the exit door.
For when it starts to grow.
Right. When the spore detects the right moisture and temperature, the new fungal hypha emerges from that pore. A pronounced snout might offer a structural advantage, perhaps directing the emerging hypha precisely toward a moisture gradient.
Or maybe helping it breach the thick, armored wall of the spore itself.
Exactly. It's a very robust design.
And what about that dense fringe of Chylocystidia on the gills? The little drumsticks, does that tie back to the weeping?
The physical architecture strongly suggests it does.
How so?
Consider the physics of guttation. If the edge of the gill were perfectly smooth, a large droplet of water would simply succumb to gravity and sheet off instantly. It wouldn't hang there as a tear.
It would just drip right away.
Right. But by deploying a dense microscopic forest of knob-headed cells right at the margin, the fungus exponentially increases its surface area.
Oh, and it creates microscopic capillary spaces.
Yes, this fringe likely acts as a physical anchor, using surface tension to hold those large, heavy, spore-filled droplets in place.
Maintaining the precise microclimate necessary for whatever evolutionary purpose the weeping serves, whether it's the bugs or the pressure valve.
Exactly. It's brilliant engineering, honestly, hidden under a very messy exterior.
It really is. Now, if the physical structure under the microscope is complex, the hidden chemistry of this mushroom is an absolute labyrinth.
It's where things get really murky.
The scientific papers transition from what we can clearly observe into some deeply tantalizing unknowns. And mystery number one involves something called hyphal crystals.
This is an area where the scientific literature simply hits a massive wall.
So the sources detail an experiment where researchers took the vegetative mycelium, the underground network, and cultivated it in a laboratory setting.
Away from the dirty soil.
Right. They grew it on agar plates and submerged it in liquid cultures, keeping it around 26 degrees Celsius.
A nice, warm, controlled environment.
Exactly. But as the mycelium matured, they observed it spontaneously forming bizarre geometric crystals directly on the hyphae.
Just growing out of the fungal threads.
We're talking needle-like structures, perfect geometric rods, and even cubic shapes. What is the metabolic purpose of a fungus growing geometric crystals underground?
Well, the formation of crystals and fungal hyphae usually points to one of three biological imperatives.
Okay, what's the first?
First, it could be mineral sequestration. The fungus might be pulling elements like calcium from the soil and locking them up as calcium oxalate to buffer the pH of its immediate environment.
Like it's building its own antacids to survive acidic soil.
Exactly. Second, it could be a storage mechanism for secondary metabolites.
Which are what, exactly?
They're complex biochemicals the fungus has manufactured, usually for defense against predators or for chemical communication with other soil microbes.
Okay, so it's storing weapons or radios in crystal form.
Basically, yes. And third, it is often a waste management strategy.
Back to flushing the trash.
Right. The fungus isolates toxic byproducts of its own rapid metabolism, crystallizing them into an inert state so they don't destroy the internal cellular machinery.
Like kidney stones.
A very apt comparison.
So which of those three strategies is the weeping widow actually employing with these rods and cubes?
We do not know.
Still nothing.
The chemical identity of these specific geometric crystals remains a complete unknown in the scientific record.
That is wild.
Researchers documented their existence, noted their striking structural diversity under the microscope, and the investigation seemingly stopped right there.
Why? Did they just run out of funding?
Probably. No one has successfully extracted these crystals, purified them, and run them through a mass spectrometer to determine their actual molecular structure.
So they could be a completely novel class of defensive toxins.
Or a unique metabolic waste signature, or a biochemical compound we haven't even categorized yet. It is a massive glowing blind spot in mycology.
It's incredible to think that in modern biology, a literal geometric crystal growing on a common park mushroom is just sitting there entirely unanalyzed.
Waiting for a curious grad student.
Seriously. But that mystery, as cool as it is, really pales in comparison to the debate surrounding its potential psychedelic properties.
Oh boy, here we go.
This brings us to mystery number 2, the Psilocin paradox.
This is undoubtedly the most fiercely debated aspect of the Weeping Widow's unseen life.
Let's lay out the evidence for everyone listening. In the world of biochemistry, there is a massive natural products database known as coconut.
The common core of neutral products.
Right. It acts as a global scientific ledger, basically tracking which organisms synthesize which specific chemical compounds across the world.
It's a huge aggregator of data.
And if you look up Lacrymaria lacrymabunda in the coconut database, it is explicitly associated with psilocin.
Psilocin.
Psilocin is the dephosphorylated, highly psychoactive compound responsible for the effects of magic mushrooms. It's what psilocybin turns into in the human body.
It's the active ingredient.
Yes. So, according to a major chemical database, this goth graveyard mushroom is hiding a potent hallucinogen.
Now, from a purely evolutionary perspective, it is actually not an impossible proposition.
Why not?
Because the Psathyrella family, where the weeping widow currently resides, is genetically adjacent to other fungal lineages, such as certain Paneola species that are very well-documented producers of psilocybin and psilocin.
So it's in the right genetic neighborhood.
Exactly. The genetic architecture, the complex metabolic pathways required to synthesize these specific tryptamines could theoretically be conserved somewhere in its evolutionary history.
The blueprints might still be in the DNA.
Right. The paradox, however, arises when you try to verify that database entry in the real world.
Because the hard proof just isn't there, is it?
No. A database entry is only as strong as the primary analytical literature backing it up.
Which means someone has to have actually tested it in a lab and published the results.
Exactly. And if you search for peer-reviewed studies utilizing liquid chromatography mass spectrometry, or LCMS,
Which is what?
it's the absolute gold standard for identifying specific molecules in biological tissue. It separates compounds and weighs them perfectly.
Got it.
If you look for LCMS studies definitively proving the presence of psilocin in a verified weeping widow specimen, you find a complete void.
Nothing.
There was no published analytical data confirming the coconut entry.
So could it literally just be a clerical error, like a typo made 20 years ago by a tired grad student entering data which then just got scraped and replicated across digital ledgers until it became accepted fact?
A data artifact is a highly plausible explanation, yes. Perhaps stemming from a misidentified mushroom sample decades ago. Someone thought they were testing a weeping widow, but it was actually a Paneolus.
That happens a lot, I imagine.
It does. However, there is a physical behavioral trait of the mushroom that kind of deepens the mystery and keeps the debate alive.
The bruising.
The bruising. When foragers handle mushrooms containing significant concentrations of psilocin, the flesh almost universally bruises a vivid, undeniable blue color wherever it is damaged or cut.
It's the classic field test.
Right. This bluing is the visual result of an enzymatic oxidation cascade, acting specifically on the psilocin compounds when exposed to air.
But the weeping widow doesn't do that at all.
No. You can cut it, crush the stem, completely damage the gills, and there is no blue reaction whatsoever. It just stays its drab, muddy brown, or just stains black from its own spores.
Right, but is it possible that the database is actually right and the mushroom is just somehow hiding the psilocin?
Hiding it.
Yeah. Could it have a unique internal chemistry, maybe an incredibly low internal pH, or an overwhelming concentration of antioxidants that actively prevents the psilocin from oxidizing, thereby masking the blue bruising completely?
Well, fungi are unparalleled masters of chemical compartmentalization. They are very good at keeping chemicals separated inside their cells.
To stop them from reacting prematurely.
Exactly. They can sequester volatile compounds in specialized microscopic vacuoles. So, speaking strictly from a biochemical standpoint, yes, it is conceivable.
It's possible.
It is possible that a tryptamine could be present in trace amounts, or compartmentalized in a way that chemically inhibits the specific oxidation enzymes required to produce that blue pigment.
But we just don't know for sure.
But without rigorous, targeted LCMS assays performed on authenticated specimens harvested from multiple distinct environments, I might add, it remains a tantalizing, totally unverified ghost in the machine.
It really challenges how much faith we put in digital ledgers. Just because it's written in a massive database doesn't mean the physical reality matches. It highlights a massive gap in our fundamental knowledge.
It highlights what I often refer to as the empty chemical profile of this organism.
What do you mean?
We become so fixated on the psilocin paradox because it's flashy. But the truth is, we know next to nothing about its baseline secondary metabolism.
The normal stuff.
Right. We don't know the chemical structure of the volatile organic compounds that give it its distinct earthy odor. We haven't mapped the phenolic compounds it likely uses to deter nematodes in the soil.
We don't even know what the crystals are.
Exactly.
Yeah.
We haven't characterized the specific enzymes it uses to digest decaying wood. The general chemistry of this incredibly common, widespread mushroom is characterized by a near-total absence of modern investigation.
Which makes what we are about to discuss next almost miraculous, honestly.
It really is a massive leap.
Because despite this massive void of overall chemical understanding, scientists managed to isolate one single specific protein from this mushroom.
And that isolated discovery has quietly revolutionized modern oncology.
This is where the story shifts from biological curiosity to profound clinical utility. We are talking about the Psathyrella velutina lectin.
Commonly abbreviated as PVL.
Now, the name goes back to one of the mushroom's old, discarded taxonomic synonyms we talked about earlier, Psathyrella velutina, but it is the exact same organism we've been discussing this whole time.
Yes, the weeping widow.
So to understand why PVL is so groundbreaking, we have to dive into glycobiology, which sounds intimidating.
It's not too bad, I promise.
What exactly is a lectin, and how does it interact with human cells?
A lectin is a highly specialized type of protein whose sole biological function is to recognize and bind to specific carbohydrates or sugars.
Okay, so it's a protein that hunts for sugar.
Exactly. To understand why this matters for cancer, you have to visualize the surface of a human cell.
Okay, picturing a cell.
Every single cell in your body is enveloped in a dense, complex forest of carbohydrate chains known as glycans.
Glycans.
Right. Think of these glycans as highly detailed ID tags or a barcode system covering the outside of the cell.
Like a barcode on a product at the grocery store.
Exactly. And they communicate vital information to the rest of the body, they say. What specific tissue the cell belongs to, its current developmental stage, and crucially, whether it is healthy or diseased.
So healthy cells are constantly assembling these complex sugar barcodes in their internal factories, primarily the Golgi apparatus, right?
The Golgi apparatus, yes, that's the assembly line.
And they present them on their surface to prove to the immune system that they belong there and they're healthy.
Precisely. Now, the PVL lectin extracted from the weeping widow has a remarkable, almost unbelievable dual specificity.
What does it bind to?
It binds incredibly tightly to two very specific letters in that complex biological barcode. Those letters are terminal N-acetylglucosamine, heavily abbreviated in the literature as GLC-NAC.
GLC-NAC, got it.
And acetylneuramic acid, which is much more commonly known as sialic acid.
Okay, so we have a mushroom protein that acts like a highly tuned magnet for these two specific sugar molecules.
Right.
Why is that relevant to cancer?
Because cancer fundamentally breaks the cell's manufacturing process.
The factory goes offline.
Exactly. When a healthy human cell undergoes malignant transformation and becomes cancerous, its internal machinery descends into chaos. One of the very first systems to fail is that delicate assembly line in the Golgi apparatus responsible for building those complex glycan ID tags.
So it can't build the barcode correctly anymore.
The cancer cell loses the ability to finish the job.
The factory starts churning out defective barcodes.
Yes. The cancer cells begin presenting what we call agalactoglycans on their surface.
Agalactoglycans.
These are truncated, severely incomplete carbohydrate chains. Because they are missing their terminal structures, they inadvertently expose molecules that are normally buried deep within the architecture of a healthy, fully assembled glycan.
Like leaving the framing of a house exposed without putting the drywall up.
Great analogy. Specifically, they expose those terminal non-reducing beta-GLC-NAC residues we just talked about.
The exact thing the PVL lectin binds to.
Exactly.
So if we think about the healthy cell's ID tag as a perfectly spelled word, the cancer cell is presenting a word with a severe spelling error. It's truncated, it's missing the last few letters.
Right.
And the PVL from the mushroom is the ultimate, highly specialized spell checker. It is programmed to highlight only those exact structural errors.
That's exactly how it functions in a clinical setting.
That's incredible.
It really is. Those specific exposed GLC-NAC residues are incredibly rare in healthy human tissue, with the minor exception of certain specialized glands in the stomach lining.
Okay, so if you find it outside the stomach.
If you detect those exposed residues anywhere else, you are almost certainly looking at a carcinoma. They are massively overexpressed on the cell surfaces of aggressive tumors, particularly lung, breast, and colon carcinomas.
So how do pathologists actually deploy this mushroom protein in a hospital? They obviously aren't sending medical interns out to graveyards with baskets to forage for weeping widows.
No. The process has been entirely modernized through genetic engineering.
Thank goodness.
Once researchers mapped the specific gene responsible for producing PVL in the mushroom, they spliced that exact gene into the DNA of bacteria, usually E. coli.
So the bacteria do all the work now.
Exactly. These bacterial cultures act as microscopic factories, producing massive, pure quantities of what we call recombinant PVL, or rPVL.
Mass manufacturing the biological spell checker.
Mass manufacturing. And in the oncology lab, researchers take this pure rPVL and chemically attach a fluorescent dye, or a radioactive marker, to it.
To make it visible.
Right. So when a surgeon removes a tissue biopsy from a patient, let's say a section of breast tissue with a suspected mass, the pathologist treats that tissue sample with a solution containing the marked rPVL.
And what happens under the microscope?
Because of its intense specificity, the PVL completely ignores the healthy cells with their fully assembled glycans. It just washes right past them.
Because the spelling is correct?
Exactly. It only binds firmly to the truncated defective glycans on the cancer cells. So when the pathologist places that sample under a specialized microscope, the cancerous regions literally light up.
Wow.
It allows them to clearly, definitively delineate the exact borders of a tumor, distinguishing malignant cells from the healthy surrounding tissue with astonishing precision.
So what does this all mean? I mean, it is hard to overstate the profound irony of this entire system.
It is very poetic.
We started this deep dive looking at a mushroom that feels almost cursed, you know? It weeps black tears. It is bitter and practically inedible.
It thrives in graveyards and disturbed soil.
Yeah. Acting as a biological sponge that concentrates the heavy metals and toxic runoff of our urban environments, it is the absolute goth outlier of the fungal kingdom.
The weird kid in the corner.
And yet, hidden deep within its unexamined, mysterious cellular machinery is a protein that serves as a precision-guided diagnostic weapon against human cancer. It literally highlights the errors in our own biology.
It perfectly illustrates the hidden architecture of the natural world.
It really does.
I mean, human engineering is incredibly advanced, but we often struggle to design molecules from scratch that possess the sheer binding specificity of a protein that evolved over millions of years out in the dirt.
Nature already did the hard work.
Exactly. The solutions to our most complex medical challenges are so often locked away in the organisms we historically ignore, or worse, actively try to eradicate. PVL proves that even a toxic weeping mushroom on the side of a polluted highway holds indispensable value.
We have covered an immense amount of ground today, and the sheer density of this mushroom story is staggering to me.
It's a lot to take in.
Let's recap for everyone. We started with the visual of Lacrymaria lacrymabunda weeping its black spores into the cemetery dirt. We unpacked the physics of that guttation, exploring whether it's managing turgor pressure, flushing toxic waste, or bribing insects with nectar.
And we watched it outcompete established microbes in disturbed, polluted soil, acting as a bioaccumulating sponge.
Right. We navigated the historical nightmare of its taxonomy, finally using DNA to break it out of the Psathyrella polyphyletic trap. We looked closely at its microscopic armor, the warty, snouted spores, and those capillary-forming sterile cells on the gills.
The little drumsticks.
The drumsticks. We hit the dead ends of its chemistry, from the unidentified geometric hyphal crystals to the completely unresolved debate over its psilocin content in the coconut database.
And we concluded in the oncology lab, where its PVL lectin is actively hunting down the truncated glycans of human carcinomas.
It is an absolute masterclass in biological complexity.
It is a remarkable narrative of discovery.
Mm.
But, as we wrap up, there is one final, somewhat provocative concept regarding the weeping widow that I think really demands our attention.
Oh, what's that?
Well, we have focused heavily today on what this organism contains.
The crystals, the potential tryptamines, the PVL, lectin.
Right. But its greatest untapped potential for humanity might actually lie in what it does.
The process, not just the product.
Exactly. We know it is a saprotroph that heavily favors the most disturbed, chemically polluted environments we create. It actively absorbs heavy metals, complex hydrocarbons, and synthetic pesticides.
From the roadsides and the golf courses.
Yes, but it doesn't just absorb them and die.
It survives.
It thrives. It manages to build complex fruiting bodies and reproduce while totally saturated in chemical toxins that would kill other fungi.
So it has to have an internal mechanism to deal with that chemical load.
It must possess a vastly understudied enzyme toolkit.
To process the poison.
Precisely. The lactases, the peroxidases, the specific metabolic pathways it uses to either break down these complex urban pollutants or safely sequester them. This is an engineering marvel.
I see where you're going with this.
If researchers can fully sequence its genome, look past the specific proteins like PVL, and truly map the mechanics of its resilience,
The weeping widow could transcend its role as a diagnostic tool for human bodies.
it could be used for the soil itself.
Wow.
It could become a foundational blueprint for advanced bioremediation.
So we could harness its specific enzymatic processes to actively digest petroleum spills or neutralize synthetic agricultural runoff.
And seal the heavy metal toxicity of our industrial landscapes. The very organism that thrives on our environmental failures could actually teach us how to reverse them.
From a weeping anomaly in a graveyard to a biological blueprint for cleaning the earth, it completely reframes how we should look at the forgotten, messy corners of nature.
It really does. Everything has a purpose.
So, the next time you walk past a disturbed patch of dirt and see a dark, shaggy mushroom crying black tears, you are looking at a masterclass in survival chemistry and profound potential.
Don't eat it, though.
Definitely don't eat it. The mysteries hidden in the soil are vast, and clearly they are worth the effort to understand. Keep asking questions, keep looking closer, and we will catch you on the next deep dive.